Chapter 4 is about bacterial growth, and so what this slide is showing us is just a variety of different media and how bacteria appear on them. So they're growing on different substrates, so different sugars, amino acids, protein mixes, and there are pH indicators in these media that allow them to change colors. And so this one right here, this is EMB actually, which we're working with in lab currently. All right. So when we look at bacterial growth, what we really are thinking about is, under lab conditions. We often model bacterial growth in the lab, and the reason for this is to help us, look at how they grow, when they grow best. And so bacterial growth is by a process called binary fission. And binary fission refers to basically taking one cell, all right, it's going to elongate, all right, it's going to get bigger, and then it's going to divide into two cells. Okay, so that's binary fission. It breaks into two. Okay, it generally will double in size, right? So this is this 2 times size thing that we're thinking about here, right? So it's going to grow in actual size. But the way we measure growth, all right, so we measure it, is cell numbers. Because we can't really see this increase in size of the individual. That's extremely difficult for us to calculate, whereas the numbers of cells we can. All right, so we can actually observe that over time. Okay? Now, binary fission, okay, all right, takes place. All right, so here we have our cell, right, and it's just getting going. All right, so it's elongating. We've duplicated the DNA. All right, so we replicate the DNA. All right, then we move the DNA, so this chromosome, into each of the 2 cells, and a wall forms in between. All right, and then the cell completely divides into 2, and the cells separate, and we have binary fission. All right, so pretty easy, right? So really, what we want to be thinking about is that you have to duplicate or replicate the chromosome. All right. We have to separate them, right? So we actually have to separate the cytoplasm and the chromosome. Okay. Let's refer to this as divide. Right, so we need to divide the 2 chromosomes, right, 1 to each cell. All right, we also need to divide the cytoplasm. Okay. All right, so those are just kind of some of the key little pieces. Now, growth in laboratory conditions. All right, we want to think of this as being in what's called a closed system, which means that we are not going to allow any additional nutrients to come in. Nothing can get out. All right, and so we call this closed or often batch. All right, and so what we're really talking about is if we think about it, we have this flask. All right, and we'll put a cap on it. And in here we have, some sort of growth solution. And what we're going to do is we're going to allow these bacteria, right? They're going to grow and replicate and grow and grow and grow and grow and grow. And eventually, right, it's going to become so crowded in here that the growth rate is going to stop. All right? And that's because we're not allowed to get, no waste can come out, all right? And no nutrients can come in. All right, so we don't allow any exchange of that stuff, and that really is going to have the effect of preventing growth from going too far. Okay, so the growth then in this kind of a system, in this batch system, which is exactly what we see in the lab. All right, what it is that it's going to give us what's called a growth curve. All right, and so if we think about this, all right, so we have an x and a y axis, all right, and we are interested in the dependent and independent variables. So the independent variable in terms of growth, right, because this is, growth is increase in number over time, okay? So our independent variable is time, okay? Our dependent variable is cell number. Now, to get this to look properly or to look right on the graph is we need to take the log of the cell number. So we have a log scale over here. Okay, so the difference between 1 and 10 and 100 and 1,000 is all the same, right? The distance. That's what it means to have it be on a log scale. Now, the way our curve is actually going to look is it's going to start out flat, all right? Then it's going to head on up, and then it's going to kind of go flat again, and then it's going to decrease, okay? And your book may show it kind of like this, all right? Now, we have what's called the lag phase, all right? We have log or exponential, okay? All right, two different names for that. Then we have stationary, which is where they've stopped. Actually, then we have death. Okay. Let me put this one in black as well, so it's easier to distinguish from the other. Okay, so that's stationary, which means they've just stopped. All right, so sort of key. So the way to think of this is the lag phase is where we're revving up growth, right? So the cells, maybe we just put them into the flask. Now they're in this nutrient-rich environment. They start to produce enzymes and they start to divide. So their division rate is slower, okay? Then it hits this exponential phase right here. And this is where they're kind of at their optimum. They're growing as fast as they can, and the growth at this point is linear. There's no change in the rate of growth, all right? So during lag, all right, so during lag phase, we get a change in rate of growth, which means rate of cell division. During the log or the exponential, no change, okay? During stationary, we're going to basically go to 0, okay? Now, I can show you this in terms of how your book shows it, okay? And so what we have on the y-axis is the number of cells. It's on a log scale. So you'll see we go from 10 to the 0 to 10 to the 2, 10 to the 4, 6, 8, 10, and those distances are the same, okay? Then we have time in hours, maybe even days or months or years. Okay, so if we did not place this on a log scale, all right, if we did not, it would look kind of like this, all right? And then it might eventually flatten and start to decline. But what this looks like, right, if you look at this red line, it looks as if the growth rate changes over time, right? So this implies, right, change in growth rate over time. However, when we look at the log scale, right here, we'll notice that this is linear, and that indicates that there is no change in growth over time. So you must plot this on the log scale, okay? All right, let's look at some data. The data, if we look here, so we've got a cell that is dividing about every 20 minutes, right? So they've sort of stacked this. So if I look at the time, whoops. So if we look at time right here, all right, the initial population we're going to start with is 10 cells, okay? So the number of generations, all right, is how many times it divides. So if the time it takes to divide is 20 minutes, we're going to be at 1 generation after 20 minutes. All right. Now, because we are actually interested in the number of divisions here, so we are at 2 to the 1, right, which equals 2. 2 times 10 is 20. Okay? Now, and so what it is our equation is actually 2, all right, to the n, all right, times n0, which is our initial concentration. Okay? And so what we want to think of then is the questions become how many generations, right? All right, how many generations starting concentration? Sorry, your starting number of cells. Okay, and we'll do some simple calculations with this in lecture, so I don't want to go into too much detail now, but what I want you to imagine is that in about 4 hours time, we went from 10 cells to almost 41,000 cells. And so why is understanding this idea of growth rate, right? How many generations, how much time does it take? Well, let's think of the amount of time that it would take, say, for some milk to add. Okay? So, if I have my carton of milk, all right, how is it they know when to set an expiration date? All right, well, basically, they know, right, if we think about time, number of cells, and it really should be the log of our number of cells, all right, they know that at 4 degrees C, all right, 4 degrees C, we're basically going to see these things go like so, okay? And they know at about a certain density of cells or about a certain length at time of their growth that they're going to create a certain amount of spoilage. All right, so that's one example. All right, another example could be just really the amount of time it takes for you to get sick after you were exposed to an organism, right? So if we think about, you're getting exposed to a virus or a bacteria, they all have some sort of, replication time, and they have to reach a certain number before you would get ill, all right? And so let's look at, right, so if we have, again, time, and we have the log of the number of cells, all right, and let's say that you become infected here, and it's going to take them a while to kind of adjust to the human body. They enter the log phase, and let's say that right about here, and let's say that this is 3 days, all right, that right about here, okay, this is when you start to see symptoms of illness. All right, and so, understanding this. The other thing we can get is we can, when disease really is going to be impacted, if you get further along is when maybe massive amounts of toxins or something. So understanding the basics of, growth is quite important. All right, the next thing we want to talk about in terms of growth is the formation of something called a biofilm. All right. And so this is an electron micrograph of a biofilm. All right, and what we're going to see then is that we've got different types of organisms here. Right, so we've got these and we've got these, and we have some fibrous solution, tissue here. Right, we've got these things reaching out. This actually is probably an immune system cell here. All right, so let me get rid of some of this stuff because I really kind of mucked up. But what I want you to see is that these are organisms growing together in a film, all right? And they can do this in your lung, on your teeth, and they particularly like catheters, artificial joints, all right, pacemakers, and they tend to grow as a film. So what does that look like, all right? And so what we have is initially, all right, we're going to start here, okay, we have what are called planktonic bacteria, free living. They find a surface that they attach. Once they attach to that surface, they start to multiply and they produce some sort of extracellular polymeric substance. So it could be a protein, a polysaccharide, and it could be a combination of those to make some sort of gelatinous gel that they can live in. Then they start to divide, and we see that we get another organism that joins our biofilm, all right? And this thing expands. They communicate to each other. They create channels that allow for nutrients to flow in and out, right? So here's a channel, here's a channel, right? And this thing continues to grow. Now, some of them return to the planktonic side of things, and they move to other surfaces to look for other places to create biofilms. Now, the biofilm is very difficult for things, particularly antibiotics. So I'm just going to simplify. I'm going to write drugs, all right? It can be very difficult for these things to get in here, right? It can be very difficult for them to penetrate into the biofilm and cause the damage that we would hope they would cause, all right? So another example of growth then is when we generate secondary metabolites. So, for example, if we look here again, we have time, number of cells, we're on a log scale, okay? So if we look over here, we've got number of cells. So we start out with the lag phase, then we get the log. So the blue line, all right, this is cell number, all right? If we look at this pink solid line, all right, this is the synthesis of metabolites. So you can think of it as concentration of some sort of chemical. So let's say that a primary metabolite, let's just say that this is an amino acid. Okay, so this is some amino acid the cell produces. This is something it's always going to produce to grow and live. A secondary metabolite, though, this might be a toxin, all right, it might be something that helps it acquire certain nutrients. It's more specialized, okay? And I want you to notice that it is generated, right? It starts being generated as we start to see a slowing of growth, right? As we see this thing flatten out, okay? And, right, that's when we start to see this secondary metabolite. Now, the reason that we cover this, right, that we're actually talking about this idea is that we want you to think about the fact that bacteria don't always produce the same stuff, right? They're going to produce different things during log phase than they are during stationary phase, right? And in fact, they're going to ramp up production of lots of things during lag, all right? And so we want to sort of keep in mind that it isn't a constant metabolic environment inside of our bacteria, and what they produce changes over time, okay? So, what are some environmental factors that influence growth? All right, so you might be thinking, all right, what would these things be? And so we're going to come up with things like temp, right? So temperature is going to impact it. If we look at, they all have what's called an optimum growth temperature. All right, and the way to think of that is, right, so if it's time and cell number, they're going to have sort of a peak. And that peak is the optimum temp. So if this is temp now and not time, and this is my number, right, at the top of that's going to be my optimum, all right? Another thing to think about in terms of temp is that enzyme rate doubles every 10C. So for every 10C increase, all right, for every 10 degrees Celsius that we increase, the enzyme activity is going to double. All right, so for every 10 degree increase in temperature, we're going to see enzyme rate double. Okay, so again, you might think, well, what would this look like, right? So we can, right, if we were to do this, all right, what would be our independent variable? Well, that would be temp, right? That's what we're interested in, and we're interested in how that impacts the enzyme rate. Okay? And so if we make some lines here, right, so if we go from, let's say we start out at 10 and we're going to go to 20, okay, and let's just say that it goes like so, and let's say that this here is 6 and this here is 12. So we've doubled in that 20 degrees change, all right? So that's really what that statement, for every 10 degrees Celsius, the enzyme doubles in rate. So, which is beneficial to the organism, right? As you warm it up, it can do things more quickly, all right? The problem is that if you get too high, what's going to happen? You're going to basically make it so that the protein doesn't function or the membrane doesn't hold together. So if you get too high, we're going to get denaturation. So you have to avoid getting too high. So if we get too high, denaturation. All right. So some things we want to think about then are refrigeration, right? So 4 degrees Celsius is what your fridge is at, right? So what we're really trying to do is slow everything down so that things don't spoil, all right? If we want to think about disease, all right, we want to think about body location. Right, different parts of the body are different temperatures. The extremities tend to be colder. The core is 37, and that may actually dictate where a certain organism survives and grows. Okay? So this table is chock full of terminology that you're going to want to be familiar with in terms of talking about organisms that grow in various temperatures and so forth. What we want to do is you really want to look at the, what is the factor that you're interested in. Okay, so for example, if we're interested in pH, all right, you really want to then have a look at pH, and you want to know, okay, we've got neutrophils, they prefer to grow near, between 5 and 8, acidophiles less than 5.5, alkalophiles greater than 8.5. And notice it's basically indicating where they grow optimally, which means where they grow best. Okay, and so we really want to focus in on that idea of where they grow best, all right, and what their limitations are. So we're going to start with the temperature, all right, as our growth influencer, all right, and we've already done this a little bit in lab, okay. The mesophiles are sort of this in the middle, all right, and the mesophiles, they are the ones that are the pathogens, all right. So our mesophiles here are pathogens. Psychrophiles are the coldest, all right, and you'll notice that they have an optimum around 10 degrees Celsius, all right. The psychrotrophs, all right, they're sort of in between the mesophils and the psychrophils. And then we have thermophiles out here and the hyperthermophiles, all right, which grow in extreme temperatures. The only organisms actually that grow above around 70 degrees or 65 are prokaryotic, all right. And the other thing I want you to get from this is I want you to look at the shape of these curves. And so I'm going to use the hyperthermophile curve just because it's out here on the edge. But what I want you to notice is that they generally are not evenly balanced. And so if I draw a line, well, I'm trying to draw a line, right down the middle of this thing, you'll notice there's actually more to the left than there is to the right. All right, so basically it's going to increase as the temperature increases, the bacterial growth rate improves to what we call an optimum. All right, and down here we have the minimum temp, and right about here is the maximum. Okay, but you'll notice the growth rate at this maximum is extremely low, and then eventually it'll hit 0. All right, so what we want to notice is that it falls off from the optimum quite steeply. All right, and the reason for that is that the what's happening there is something, either the membrane or a key protein, something is becoming denatured. All right, and so what happens then is the growth really plummets at that point, and so we don't have a normal curve. We have something that is much steeper on that side. All right, so what's going to happen in general is that the growth increases with temperature, all right, which is what we see if we look at the thermophile here. It increases, and then something's really going to go wrong, and the growth is really going to just fall off a cliff, more or less. Okay? The next thing we want to look at are oxygen requirements, and we're going to do this by looking at where the organism grows in a test tube. All right, and so if we have our media here, okay, it's inside of our tube, all right, we're interested in where they grow. And so, for example, if they only grow near the top, right, so they just form a band and we get this dense growth here, this is where oxygen is present, right? So if we think about oxygen, all right, in the tube, in the airspace of the tube, we've got the oxygen, but deep down in here, right, down here at the bottom, we're not really going to have much oxygen. And so what we want to do is we want to think of this thing as being sort of aerobic at the top and anaerobic at the bottom. Okay, and so, oops. All right, so if we have this aerobic area at the top and it only grows there, we call it an obligate aerobe. All right. If it were to only grow at the bottom, all right, so let's say it's only down here at the bottom, it would be an obligate anaerobe, right, requiring the absence of oxygen, okay? And if it grew throughout, right, so now we have growth everywhere, all right, at this point we call it a facultative. Oops. All right, so we call it a facultative anaerobe if it grows in both oxygen and without oxygen, right? So if it grows in aerobic environments and anaerobic, so facultative anaerobe. Now, the main difference between these organisms is their ability to deal with different forms of oxygen. So when we use oxygen in metabolism, right, so if we're using the oxygen, we're going to generate things like O3 radicals, all right? We're also going to generate hydrogen peroxide. Sorry, that was my phone. So we're talking about hydrogen peroxide. And so we need to get rid of these things that form when we use oxygen as a terminal electron acceptor, all right? And so basically if the cell's doing cellular respiration, it's using oxygen, it's going to generate superoxide, and it's going to generate hydrogen peroxide. And we need to be able to deal with these. The obligate anaerobe cannot deal with them. The obligate aerobe, the facultative anaerobe, they can. All right. So so if we go back here, we're interested in this idea of the superoxide and the H2O2, right? Which of those will actually be has this nice figure here. Let me grab it. I need to get it in my view. All right. And so what we'll notice is that we've got our obligate aerobe first. All right. So right here. Then we've got the facultative anaerobe, the obligate anaerobe. Now we have a couple of other organisms, the microaerophile and the aerotolerant anaerobe. All right. So what we want to notice is where do they grow. So the obligate aerobe grows at the top. The facultative anaerobe grows throughout, but you'll notice it's denser at the top. All right, so it grows better at the top where there's oxygen present. The obligate anaerobe only grows where we do not have oxygen. The microaerophile actually grows where we have low oxygen. So notice it's not at the top. It's a little bit down from the top. And then the aerotolerant anaerobe grows mostly throughout, but is going to prefer the bottom. Now, the key to this is an enzyme called superoxide dismutase. All right, so we have superoxide dismutase and something called catalase. All right, so the obligate aerobe has superoxide dismutase and catalase, okay, which means it can deal with O3, right, so the oxygen radical, and it can deal with H2O2, not a problem. All right, it has those two enzymes. The same thing is true for the facultative anaerobe. It will have both superoxide dismutase and catalase. The obligate anaerobe is negative for either, so it doesn't produce either of those enzymes, right? So it can't deal with the presence of oxygen at all. All right. The microaerophile will produce some, all right? So it's a low-level producer of superoxide dismutase and catalase, all right? And the aerotolerant anaerobe produces superoxide dismutase, but not catalase, all right? And so basically, this allows them to either exist in the presence of oxygen or not, okay? So if we... All right, so we want to focus then on the presence of these two things, whether or not we can deal with them. Okay, so make sure you do know which of these have these enzymes. So you want to focus on the obligate anaerobe, has both, facultative anaerobe has both, obligate anaerobe has neither, microaerophile has some of both, and the aerotolerant has superoxide dismutase but not catalase. All right, so you want to keep track of all of them. All right, so our key enzymes are catalase and superoxide dismutation. So you can imagine then a question that would look a bit like which enzymes does A, and let's say, it's a, and so it could be an obligate anaerobe, for example, and the answer would be neither, right? And so what if it was a microaerophile? Well, some of both. So we want to, you want to be able to answer this question, all right, when you're done with studying that table that we just went over. Okay. All right, up next is pH, right? So if we want to remember what pH is, right, it's the negative log of the H plus concentration, all right? And what we're interested in is at what, we want to answer the question, what pH does my organism grow at? All right, and so that's really our question here. And so what we want to do then is we want to look at the fact that we've got a number of organisms that can do this. Okay, we've got various ones, and so we want to draw a graph and have a look at them. All right, so we want to draw our graph like so, and so down here we're going to put pH, okay, and over here growth. All right, so if I put 7 here, right, and so I go 6, 5, 4, 3, 2, so like so, and we'll go 8, 9, 10, okay? We'll go ahead and put 11 on there. All right, so the first group, all right, are the acidophiles, all right, and they're going to start around 5.5, and they're going to exist best in environments that are below that. So I'm going to put my acidophiles here, right, so acid-loving, all right. Then in the middle, all right, so from about 5.5 up, right, probably to right around 8.5, right, so these are a little different. That's a terrible line. Okay, so I'm going to make my middle right here. Okay, and then the last one is going to be my alkalophile, and they are going to be more up here. Okay. Now, in this case, we want to keep in mind, so we've got acidophiles, mesophiles, or I guess in this case we want to call them neutrophiles, sorry. All right, so we want neutrophiles and alkalophiles. All right. Now, I'm not that big of a stickler in terms of you knowing exactly what their pH ranges are, just kind of having a basic idea as to, we're really talking about 8 and up for the alkalophiles, around neutral for the neutrophiles, and then below 5.5 for the acidophiles. Okay? But there is a difference. When we looked at the temperature, all right, when we drew this graph in, it was not normal in terms of to the right of the center was smaller than to the left, but now it should be equal. All right, the shape of this curve should be symmetrical, and that's because an increase or a decrease in the H plus concentration has the same effect on the organism's ability to grow. All right, whereas temperature, as it goes up, once you hit that you denature something, it really destroys it. Okay. And so in this case, we have to think of it in terms of the presence or the absence of H plus can actually influence the shape of a protein kind of equally. All right. So next up, we want to talk about water availability. All right. And so this is really something that you have already got a feel for, right, in terms of osmosis. So if I have a cell, right, and on the outside of it, I have an awful lot of this solute, whatever it is, and very little of my solute on the inside. Right, we all know that water is going to tend to move out. All right. The other option is that we could have an awful lot of solute on the inside and very little solute on the outside, right? And that would then, if I get rid of some of these, now we know that water is going to tend to move into my cell, okay? And so we've been doing this in terms of osmosis, okay? So what we want to really, in terms of sort of defining organisms, is that we have bacteria then that are halotolerant, or so we have halotolerant and halophilic. To describe organisms that basically this one, right, will tolerate high levels. In a common example of this is S. aureus, and so it can handle high levels of sodium chloride. Okay, halophilic require high levels of salt. Okay, and so some examples of things are, like if we think about jelly, for example, there's so much sugar in there that there's not very much water available, and very few things can actually grow on it. All right, some fungi can, which is why sometimes jelly in the refrigerator will become moldy. But at the end of the day, most of the pathogens that we encounter they can't grow. They don't require high levels of salts. S. aureus is a pretty common pathogen, and it is tolerant, and we'll use the presence of salt as a way to select for it. All right, and so it's kind of a good thing to realize that the halotolerance then is something that we want to be able to use as a way to detect the presence of certain types of organisms. Okay? All right, so this is just kind of going over the diagram that we just looked at. And so if we look at this, in this case, we've got a lot of dissolved solutes on the outside, the water exits, and this organism here, it shrivels up. Okay, so the question would become, how would we adapt to these conditions, right? So in terms of adaptations, what we want to do is we want to look at, what is the environmental factor? So is it temp? All right, is it pH? Is it solutes, right? So salt tolerance. Is it oxygen? All right, and so what we're going to do in class on Monday is we're going to look at how these organisms, right, can grow in different environments and how they can survive them, right? And so we'll actually we want to put these things together in a manner that lets us look at an organism from its capability, how it's adapted to this environment. Okay? And